I want to thank Martin and also thank Aubrey. It's great to see you. Now, I think that you said you were in stealth mode. The one thing we don't need in this field is Aubrey in stealth mode, so it's great to see him back. There's no place to sit my coffee. And so I'm going to tell you some thoughts today about how we translate Healthspan and lifespan. This is something that I was kind of just a basic biologist trying to figure out mechanisms of aging, and then at some point decided maybe we should do something about aging. And so this is some of the thoughts that I've had along the way about what we might want to do. And so what we're doing at NUS, I would say, is creating a full program. We have a lot of basic science and preclinical research, translational research. You heard about some of the clinical science yesterday from Andrea, and I'll tell you a little bit today. And then also driving sort of longevity science in the field. And we do that through the webinar, lots of education things, trying to teach government people about healthcare, et cetera, et cetera. I think some of the facets of this that we're really focused on is, one, being agnostic. So when we look at interventions, we all have our favorites. None of us are unbiased, but we're trying to look at a range of different interventions to target health span and compare and contrast which ones work in what context. We're trying to be very collaborative, and that means working with other academics. If you have things you want help testing in mice or various other places or humans, come talk to us. Happy to collaborate on that, and also work with companies. There's several that we're already working with. And I think the bottom part, as I said earlier, is we got to get human. I think that the field has learned a lot. I can show you pillars and hallmarks of aging and talk about them, but I think the main part is that if we're going to reach the world, we have to do that by generating human data that's really compelling. And we've talked a lot about regulation yesterday, and that's important to deal with regulators, but what I'm more interested in right now is just generating compelling data in humans, because I think if we can do that, we can really make a case that will resonate much better. In our preclinical studies, we have a lot of different systems, yeast, worms, and flies. We haven't gone away from them completely. We're working with a couple labs at NUS who study killifish aging, and I'm getting more and more interested in that. Organoid models, my lab and others. Mice, mice were a bit vilified yesterday, so I thought I'd mention that. And bioinformatics, a lot of AI people, and I'll tell you a little bit about some AI today. One of the things we're doing, and Andrea alluded to this in the human studies, is trying to match up biomarkers of hallmarks and pillars of aging with systemic biomarkers of aging. I think we don't really understand to what extent different hallmarks are contributing to these newer biomarkers like methylation clock, and it's something we need to disentangle, so that's something we're trying to do. And so I thought I'd have one slide in defense of animal models. Nobody thinks that mice are the only thing we should work on, and actually I'm very supportive of most of what was said yesterday about getting as much human data as possible. I'm also supportive of medical tourism for people that are educated and want to make those decisions. I wish we could find a way to consolidate data from this medical tourism so we could really look at what's working and what's not. But you heard a whole panel of people yesterday talking about different approaches, and I don't think it's an either-or situation. I think that we should be pushing the envelope in different directions in this field, and I'm not sure which ones are going to be the most important down the road. But back to mice. So, a lot of the problems with mice are people say there's replication issues when some pharma company tests 50 mouse reports, and then they say, well, we can only repeat 6 of them. Or you do mouse studies and they don't replicate in humans. So there are those problems. First of all, the pharma companies take one shot at replication of something, and it doesn't work exactly right, and then they move on, which is fine for them. But having tried to replicate a lot of data and having published a lot of negative data in my career, which created some fights, I have to say that if you want to really determine whether something can be replicated or not, you have to try hard. And we put a lot of effort, for instance, into looking at resveratrol or other things that we've done. And in some cases, it took a lot of effort to replicate that. So I wouldn't run away from this model. I think replication is critical, and that's what we should focus on in animal models. I don't think we should disregard them, though. I think they're still quite valuable. I think one of the big challenges is people don't use age-appropriate mice, and this has sort of been a mantra that I've been on for several years. If you want to study Alzheimer's disease, don't do it in a young mouse, do it in an age-appropriate mouse, and then maybe you'll get results that translate better. Also, I think it's better to look at natural diseases that the mice get rather than create artificial diseases. And so people will tell you mice don't get Alzheimer's, we can't study that. Mice do get neurodegeneration with aging. And if you want to understand the aging contribution to neurodegenerative phenotypes, maybe you should look at the aging mice and normal neurodegeneration that happens in those mice. And what you learn might be translatable in ways that people are not thinking about right now. So, and finally, there's just limitations to working in humans. We can do sort of cause-effect experiments and take a very mechanistic approach in animals that's still hard to do in humans. So I think we need both of these approaches. I'm not trying to say we should only do animals. And so we've changed our mouse approach considerably. We're not doing survival studies in mice anymore. So we do interventions starting at 18 months of age, following on what the ITP program does in US. And then we do 6 to 9 month interventions. So that's a late middle-age animal, and then at 6 to 9 months it becomes frail, and then we can look at whether our interventions prevent frailty, and we can use a number of biologic aging markers that I'll talk about. The reason I think we've done this is, one, it saves a lot of time and money. It allows us to test a lot more models, or a lot more interventions. And second of all, it tries to align what we're doing with the human studies, which are 6 to 9 month interventions looking at biomarkers as outcomes. And I think by iterating back and forth, we can optimize the mouse to teach us about what's happening in us. And of course, we want to get human. You heard from Andrea yesterday, and I think there's some key things that have to happen. We need to validate things, the interventions that validate them in humans, see what's working for which biomarkers and which physiologic phenotypes. And ultimately, I think we need to move toward optimization and personalization. No intervention is going to work in everyone. Exercise won't work in everyone. At least if we prescribe the same exercise, it's not going to work in everyone. And so I think that we need to understand who's going to respond to what interventions. And right now, we just don't have a feel for that at all. And I want to reemphasize what was said yesterday, is that we want to do everything academically, at least, that we can scale. Not just to wealthy people. It's not about billionaires, it's about billions. And what we'd like to do are have interventions that target Healthspan in the Singapore population. So we look at the whole island as sort of a test bed, and let's make this a place where we can really push Healthspan and show what's possible. So it has to be cost-effective interventions and cost-effective biomarkers. And so we use a whole range of biomarkers. I'm not going to talk much about them today. And we're looking at healthy or early pre-disease individuals and a lot of different interventions. So it's really sort of a mix and match approach right now. And we have a number of studies ongoing. We're just finishing a really deep phenotyping using a lot of clocks with a number of 450 people from Singapore of all 3 ethnicities. And so that should be sent out hopefully in the near future. We also have a collaboration with the National Precision Medicine team where they've done genome sequencing on 10,000 Singaporeans, and we now have methylation clocks and telomeres and other things on those 10,000 people. Interesting things there. I can't talk about it because it's a large collaboration that I promise not to say anything yet. I don't like that, but I have to do that in this case. I will say that the aging rates are going to be different among different ethnicities, which maybe isn't that surprising, but I think it's really interesting data. And then we mentioned intervention studies, first one AKG, hopefully rapamycin's the next one, and maybe 5 to 10 more in the near future. And we have a lot of different collaborations and cohorts going as well. So what about interventions? Let's go back preclinically and look at what we're doing to try to be at the front end of the translation. There are a lot of potential interventions out there. There's no real shortage of these. If you look at the drug age database, there are 1,000 distinct drugs that are reported to have some effect on aging. There are 110 of them in mice. And so it's not like we are short of ideas. We need to get better data in both mice and humans and figure out which ones are working the best. And we've sort of taken that approach in my lab now, where we're all of those molecules on the left we're looking at in different kinds of studies. And I think that that may not be the exotic approach in the lab, but I think that we need some place where we can compare the different ones in the same environment, in the same mice, in the same context. So I'm going to talk a little bit about alpha-ketoglutarate and Gemfibrozil today. Those other things in green, urolithin, rapamycin, et cetera, are things that we see beneficial effects on health span and lifespan in mice. They reduce frailty or extend lifespan, and I'm quite excited about a number of them. Particularly, we're working on glycine right now. Urolithin looks very promising in our hands. So I'm not going to talk about those today, but you're welcome to ask questions on that. I'll first talk about alpha-ketoglutarate, and I show a little bit of data that's we've shown in the past, so I'll keep this short, but there's a couple points I want to make. It's a collaboration with Ponce de Leon Health, and it's Tom Weldon's in the audience today. You're welcome to talk to him about everything PDL. And what they came to do is support funding at the Buck Institute originally to look for combinations of natural products that would have beneficial effects on health span and lifespan. And I'm not going to tell you about all the combination data. I'm just going to focus on the main ingredient that we found from this, which was alpha-ketoglutarate. So we tested a whole range of different things in worms that were thought to affect aging, and then went to mice. And AKG was, I think, the clear winner among the things we tested. So I'm going to tell you a little bit about a mouse experiment today, where we started the intervention at 18 months of age. In this case, we let the mice go to natural death. Looked at health span, looked at frailty, looked at a bunch of age-related parameters. I'm not going to show too many slides, but the paper is down there. You can go find it. Just one slide on the mice. This is looking at frailty by age. And you can see, maybe I'll point to this. You can see that the frailty in the control animals in blue goes up quite a bit faster than the frailty in the alpha-ketoglutarate treated animals. This is months of age after the intervention. The problem with looking at it this way is that animals are dying along the way. So if you take this animal that has very high frailty, at 23 months, it's dead by 25 months. And so that makes the analysis a little bit hard. So what we did after all the animals died is recalibrate the frailty measures to the percent of the lifespan of the animal when we took the measures. So 50 to 60, 60 to 70, 70 to 80, et cetera. You can see the frailty goes up pretty steadily in the control animals, and it's about 50% reduced in the alpha-ketoglutarate treated animals. This is females, it's also true in males. And so what we want to argue is that this compound compresses morbidity. It extends lifespan by a small amount, maybe 10%, but it has a dramatic effect on health span. And if that were true in humans, that would be a great molecule. So that's what we've been trying to test. And we reported last year a study that is not a placebo-controlled study. This is just users of a product, Rejuvent, by PDL. And Rejuvent is AKG plus low-dose vitamin A if you're a man, or low-dose vitamin D if you're a woman. And these are 42 participants in the study. They took the product for about 7 months, and we did methylation age before and after. Now, we're going back and doing placebo-controlled studies. So I take this for what it's worth, but I think it's interesting data, and I'll tell you why. This is the change in biologic age on the left. And going up here means that your biologic age is going down. And so we saw about a 7-year reduction in biologic age on average from people that took this product for 7 months. Do I think there's a placebo effect on biologic age? Yes. I think if you're paying $100 a month for a product to be younger, you're younger. But hopefully not 7 years. I think it may be a very small effect. So what I think we're looking at here is a combination of the effect of the product and maybe some placebo effect as well. And that's what we need to sort out with the future studies. But I think the interesting thing here is that there were parameters that defined the response for people. And two of them were people that were older chronologically responded better, and people that had a biologic age that was older than their chronologic age responded better. So essentially, this compound was helping couch potatoes, or people that were not aging so well. Will every compound respond that way in humans? We have no idea. I would have even guessed this. After I showed this data, everybody said, oh, that's obvious. I didn't think that going in. In fact, AKG is something that people that do a lot of exercise take to enhance their exercise performance. So it may well have been the fact that the people that were out exercising and biologically younger would respond better. That's not what we saw in this context. Now, there's another study going on in Indiana where we're analyzing the data. This is a placebo-controlled study sponsored by the company, and we're looking at the data, so it's a Rejuvenant product. And we kind of partially shot ourselves in the foot with this study because we set this up before we had that other data. And so what we selected for the inclusion criteria was 45 to 65 disease-free. We want healthy individuals for this study. If you do that in Indiana, and then you look at the biologic age of those individuals who are disease-free in middle age, it's about 4 years younger than their chronologic age. And so we inadvertently picked the people that are least likely to respond, I think, to AKG. And so we're doing another study in Singapore now, and with time-release AKG, and what we are using as inclusion criteria is people that are biologically older than their chronologic age at baseline. Is that the right way? I don't know. I think the point I want to make, though, is that we have to start doing more and more of these studies, and I don't think any of us know exactly the right approach yet. It's a bit of a learning process. And so we have some signals here. Hopefully we'll be able to tell you about them soon. But I think we'll get a bigger signal if we look in the people that are not aging as well, at least in the context of AKG. Will that be true with other molecules? Yet to be determined. Gemfibrozil is an old drug. So this is new tricks for an old drug. I want to show this data, though, because I think it's a really ideal candidate to think about for translation in Healthspan. And we're looking for ways to get support to do that, actually. We originally found this drug in yeast, and I was kind of skeptical of screening yeast for drugs. But it turns out that we had a trick to do that with a collaborator, Michael Palominas at Texas A&M. And we found a number of drugs. We reported a paper on ibuprofen. This we haven't published on Gemfibrozil, and you can see that it extends lifespan in yeast and worms, and it affects Healthspan parameters in worms as well. I'm not going to show any slides on this, but Matt Kaberlein talked at the ARDD meeting, and we had a little bit of discussion about this yesterday. I think that we have just barely touched on the intervention space for aging at Healthspan. And so what Matt and Jan Gruber and I have done is start a company now to do massively high throughput screening of worm aging and worm disease phenotypes to try to screen a lot more of the drug space. I think there are some good candidates for intervention in Healthspan and lifespan, but there's a whole universe out there that we don't understand, and particularly when we start to combine things together. Whenever we test combinations in mice, I have no way of predicting the outcome. And a lot of times they counteract each other. So we need a much bigger data set on how to combine things together. And I say that also because there are a lot of people out there taking 10 or 15 different supplements at the same time. I have no idea what that's doing. I take no responsibility if anybody's doing that. So we need to learn more. But Gemfibrozil is an interesting drug. It's a fibrate, or at least it's called a fibrate. It doesn't look like other fibrates, though. This is the structure. And the data is quite good. This is really old data from Park Davis showing a reduction in triglycerides and an increase in HDL for people with hyperlipidemia. So it's still a highly used drug, sort of secondary to statins for hyperlipidemia and used directly for hypertriglyceridemia in some cases. And so we're curious why this was affecting yeast aging. Now the mechanism, this is work by Chang He and a great graduate student in my lab now at NUS, Charmaine Tien. And it's supposed to go in and bind to this complex, which contains PPAR-alpha, release the complex, allow PPAR-alpha to go to the nucleus and activate transcription. Now, the aging phenotypes don't require PPAR-alpha, so we didn't think that was the mechanism. And the other fibrates don't affect aging in our hands, so we think Gemfibrozil is doing something entirely different. And honestly, if you go back and look at the data, I don't believe that PPAR-alpha is a target of Gemfibrozil at all. I think it was convenient to call it that, because you could call the drug a fibrate and get it approved faster, but we can find no evidence of that. And so we've been looking at what Gemfibrozil really does. And in yeast, what you can do is you can ask yeast strains to grow on all kinds of different carbon sources, and then ask where the drug causes sensitivity. And what we found is that when you look at dipeptides and tripeptides as carbon sources, the yeast can't grow if Gemfibrozil is present. This is a very elegant way of showing that, because you can take a toxic dipeptide that when it gets into the cells, it kills the yeast. And so you have this zone of inhibition around the drug. But when you add Gemfibrozil, you block that toxicity. And what's happening is it's blocking import of these dipeptides and tripeptides. And when you can look at the yeast, you find there's a dramatic reduction in amino acid levels in cells when you add the drug. That's true also in human cells, and we can show that dipeptides don't get into the worm. It's a very consistent phenotype across different species. And so I'm not going to talk too much about Gemfibrozil today, just to maybe tease you a bit. I will say that we've done a lot of complicated intestinal uptake experiments now. And what you find is that dipeptides get taken up quite quickly in the intestine, but if you add Gemfibrozil, you can block that uptake. Interesting, losartan has that phenotype as well. That was our positive control. And I'm curious to go back and look at the aging phenotypes of that drug as well. And so we're pretty confident that's what's happening. And if you block amino acid uptake, what you end up doing is inhibiting mTOR activation. So David Sabatini's done a lot of work on how amino acids activate TOR. And so we could test that. So you have starved cells. They have, looking at a substrate of mTOR, very low phosphorylation. You add amino acids, you get phosphorylation of mTOR, and Gemfibrozil doesn't block that. You add one amino acid, leucine, you get the same effect. But if you add dipeptides, you activate TOR, and Gemfibrozil blocks that phenotype. So it's pretty clear that that's what's happening for aging. And I think what I'm going to do is go on, because I have very limited time. But the point I want to leave you with is that we know now the targets for Gemfibrozil, I think both for aging and for hypertriglyceridemia. And so that puts us in a place where we can now derivatize Gemfibrozil and actually optimize it for the real targets of the drug and not PPAR alpha. So one more thing real quick. I'm just going to touch on this. Max, somewhere, the guy in the hat, working on lipids. He talked a little bit about generating a lipid clock yesterday. So we have a lipid clock now. We have one in worms and mouse brain. But we got interested in lipids because these are very bioactive molecules, and they're very poorly understood. If you go to the lipid database, there are about 25,000 lipids in it. Sorry, that's the summary of what's happening with the lipid clock. Talk to Max about that. I don't have time. If you go look at lipids, there are about 25,000 of them in the lipid database. And we only measure 100 to 1,000 or so when we do lipidomic studies. And so there's a lot of unknowns out there. And these lipids are very active, and they're predictors of health. They're good biomarkers. So we wanted to go back and look at lipids. And so what Max did is an artificial intelligence approach trying to classify the 25 lipids. And so this is a classification scheme. And you can see that we can separate different kinds of known lipids into the known classes. Different colors are different classes of lipids. And we thought this will be really interesting because we can find the nearest neighbor of any lipid you're interested in and try to find other molecules that have similar activity. And we thought, let's do a control. The control was to look at other molecules that are not lipids and make sure our lipid classifier doesn't call them lipids. So we went to the PubChem database to do this, 110 million molecules. And you get something like this. This is 250,000 random molecules from that database. The colored ones are the lipids we did before, and a lot of them don't classify as lipids. That's great news. But if you zoom in, you'll see that, like, if we look at corticosteroids here, there's a lot of gray ones in there. And in fact, the PubChem database is full of lipids that aren't being classified as lipids. So in other words, we know about 300 of these lipids in the known database. But we found 30 more out of 250,000 from the PubChem database, and really that means there's 15,000 in that database that are unidentified. So what that means, there's a whole world of lipids out there that are really hard to access because even though they're known about, they're not classified in any kind of way. And so what we've done now is match that to PubChem terms, and now we can take any particular lipid you might be interested in, take convalitoxin, for instance. This is cardiac glycoside. And inhibits this ATPase. And we can just look and find a whole group of molecules that look a lot like this. Some of these might have been tested by companies. Some of them haven't been. And so we think this is a great database now to really go in and find new molecules related to anything you're interested in the lipid database. I'll just show one more. This is 17-alpha estradiol, which a lot of people know in the aging field extends lifespan in male mice. Here's a bunch of other molecules that look something like it. So there's a lot of other lipids out there that you may know about, and we can do this with any of them at this point. So the conclusion from this part of the study is that by clustering lipids, we found that there's not 25,000, there's about 2 to 4 million known lipids, and we have a much easier way, I think, now to work with them. So that should lead to a lot of new molecules that have interesting phenotypes. And this, we can link them to the literature. So if you're interested in a particular disease or mechanism or pathway, we can now use that approach to find the right lipids to study. And we're also using generative approaches now, so we can take a lipid and make new molecules that look like that lipid that haven't been identified before. So I think hopefully we can open up this class of molecules a little bit to more testing, because they're really exciting, as I said, bioactive molecules. And we didn't really plan to go down this path, I think, but Max has really taken a smart approach, and I think this database will be really useful. Okay, so just a couple things to conclude. I'm running over time. Sorry I rushed through the lipid thing, but first of all, we not only have a healthy longevity program at the NUS, we also have, thanks to Nicole Shanahan, an Asia Center for Reproductive Longevity and Equality, which I wanted to call All About Ovaries, but I was told I couldn't do that. But we're doing a lot of work both in animal models and in humans, because we have an IVF clinic at the hospital to look at what's going on with ovarian aging. I think it's extremely understudied and important for aging, so we're excited to be working on that. And then finally, Andrea mentioned this yesterday, we have this longevity webinar. You can go to this site. It's free, and register if you're interested. There are about 65 talks now that are online and available if you want to find your favorite person, including Aubrey or a lot of other people that were at this meeting. We're really trying to bring together not just the scientists, but the clinicians, the investors, and anybody else interested in healthy longevity under one virtual roof, so that we can address the key issues in extending health span. So I'll probably reach out to some of you to do the show. Somebody came to me and said, you've done 62 shows and I've seen 61 of them. And my thought was, what's wrong with this person? But actually, they've become quite popular, and some of them reviewed over 100,000 times online after the show. So hopefully it's having an impact. So with that, I'll stop. A lot of people have helped out. It's great having Andrea at NUS. We're very different people in terms of how we think, and I think we're managing to make both of us better by working together. So it's exciting to collaborate with her, and I have a couple minutes to take questions. Thanks for inviting me.
Very inspiring talk, Brian. And the data about AKG, it was impressive. What's your understanding of the mechanism? How is it that this is? Do we have any clue on the mechanism, or are there conflicting theories?
Yeah, this is the problem with natural products, and you can make the same case with NAD, is they participate in hundreds of different reactions in the cell, and you're trying to figure out which ones are most important for aging. And I think that a lot of these interventions actually are not doing one thing. They're targeting this network that the outputs of which are all these hallmarks and pillars of aging. But what they're really doing is preserving this sort of health span network in your body that's reacting to things that are happening with aging, compensating and keeping you disease free. And so I think that a lot of these interventions are preserving network function. The problem with saying that is you don't know really what that means. AKG is a substrate for demethylases, so that could be affecting methylation clock directly. We also think there are really cool things happening in the microbiome when we give mice AKG, and it also participates in a number of reactions that generate interesting molecules. So we're looking at where it's going in the body, which is largely liver and kidney, and looking at phenotypes it has in those cell types to try to get at mechanism, but it's a challenge. I can tell you a lot of things it does do. It's really hard to say which ones are important for the longevity phenotype. So,
yeah. Martin Sieber, thank you very much for this talk. I had a question and a comment, and now I realized I have two comments actually. So number one, I do mouse work, and of course we had our failures, and one is that it's not only the age, but it's also the background. And for instance, age-related hearing loss, we studied at the Cure Clump, and the CAS background. And we studied emphysema, smoke-induced emphysema, and we had it on a black 6 background and was entirely wrong. So I think a list, it's out there in the literature, but we need a list really saying, if you want to study, for instance, emphysema COPD, then you have to have a AKR or something. The comment, I also wanted to ask for the alpha-ketoglutarate. It's needed for hydroxylation and actually impairs HIF signaling and has some antagonists, like for instance succinate and fumarate, and it requires iron. So I was just wondering if in some of your non-responders you had a high upregulation of other TCA intermediates, and or if these mice, for instance, did not show the HIF or still had the HIF signaling active. And then you would know where the failure comes from.
So the first question, I agree with the comment. I think the question on TCA cycle components is really interesting. There have been studies in worms showing that adding other TCA cycle components extend lifespan. And we haven't tested those in mice. It would be great to go back and compare them side by side. Certainly adding AKG is probably driving elevated levels of other ones, at least in the cell types that AKG gets in. So that's a really important thing to look at. And then we haven't looked enough at HIF-1 alpha. Another potential mechanism is regulation of HIF-1 alpha through prolohydroxylases. And that's another candidate for how AKG could be affecting aging. And I honestly can't answer whether that's important for the phenotype or not. I think it's a great question. So.